Thanks very much. I'm delighted to be here. This has been a really excellent conference. As Ken said, I have many hats, but mainly I am just a mom. And my entire interest in this area comes from the diagnosis of my two children, Elizabeth and Ian Terry, in 1994 with pseudoxanthoma elasticum. PXE means never having to say pseudoxanthoma elasticum. The kids charge me royalties, and I have their informed consent for using their photograph in my talks. Like all parents kind of faced with sudden illness, 2 days before Christmas in 1994, my husband and I found out our children might be blind, they might have severe cardiovascular disease and other ramifications that we didn't expect. They were quite young. And so we did the thing that we usually do, which is to kind of go into overdrive, and we surveyed the literature to look for what could we understand about PXE. And in those days, it was very little, but still some. We then followed the literature to the researchers and interviewed the researchers. And then we investigated these issues with the consumer community in the form of Genetic Alliance, which is a coalition of disease groups. The biggest and most important thing, though, we did was survey the literature. That was a really key piece for us, which led us on another kind of journey. Visiting Countway's library cost us $25 a visit. We were just average folks living in a suburb of Boston and not able to pay that much money every time we wanted to go survey that literature. In those days, you couldn't get anything much through internet, Grateful Dead, med. We then volunteered at the Brigham and Women's Hospital, because if we volunteered, we could get in free. But it was hard to spend the time we needed to do research on the disease and also volunteer and also do our full-time jobs, and so instead we mainly stole passwords. We found researchers and others in the community who would let us borrow their passwords or figured out ways of trailing med students into the library, and we were able to get the literature that we needed. With the literature that we needed, and I'll talk also in a few minutes about public access because that's a major passion of mine, we were able to put together a diagram. My husband was in fire protection engineering, and so 2 months after the kids were diagnosed, he put together this diagram as a map for us to follow for our research, and it's actually done us good till today when we have projects going on in almost all of these areas. Because of our work, including some wet bench work at Harvard, we were able to be part of the gene find, the gene discovery, and published 2 back-to-back papers in Nature Genetics, getting the 2 competing groups to work together so that we could publish back-to-back. And perhaps I'm the only person with a master's in religious studies to publish 2 gene articles in Nature Genetics. We went on to patent the gene. We did that so that we would remain stewards of the gene and be able to leave the gene in an open access commons kind of environment, and so that no one else would lock it up. We were criticized in a number of corridors for that, for interfering with the science, but in fact that's been quite useful to us in order to keep us in the mix coordinating the disease research. We built a BioBank, and that's what I'm going to talk to you mostly about today, in as much as it dovetails very nicely with Genbank. But basically what we looked at there was how are we going to take a commodity like blood and tissue, realizing that many people told us that you cannot herd scientists, they're like cats, and we said, well, you can herd cats by moving the food, so we could herd the scientists by moving the samples. We took all of the samples and clinical information and aggregated them on one side of the firewall, which took care of many of the messy issues, and I'll go into detail on some of those when I talk about the BioBank. But this early form of the BioBank allowed the research enterprise to chug along quickly, and the participant and donor needs to be taken care of well on our side of the firewall. We were coming into this whole thing at a very good time. So 1994 puts us 10, 12 years into Genbank. Databases were becoming more common. The internet was becoming more useful. And maturing patient advocacy scene overall. So, and I'm not going to go through this in any detail, but you certainly all know the stories of the early AA and the early CF Foundation, TASACs, et cetera. And when we came in, there was a lot of patient movement, patient power sorts of stuff, fueled by biobanking beginning, internet advocacy starting. And then we're really looking into coming into a decade where I think we're going to see a lot more of the commons, a lot more of sharing of resources so that we can get to the translation and delivery that both Francis and Betsy talked about eloquently earlier. I think the other thing we're seeing is a shift in business models generally. And if we look at the industrial age as the old model and the information age as the new, we see a lot of differences about how we're using the materials we have. And in fact, in this age, with information being the material, it's not scarce, and it's not hierarchical, and it's not linear. It's much more organic. We're seeing a lot of win-win, a lot of network sorts of collaborations going on. And so we've come into this at a very good time when we can actually leverage some of the forces that are at work just in the general society as well as in science. What matters to us and what we keep focused on, of course, is improved health outcomes, and the pathway to that certainly has many pieces, including lots of the ones we've seen over the last 2 days. We really want to stay focused on health outcomes and have that as our driver and keep that overarching perspective. At the same time, we know there is no Delhi number, so nobody is going to call, as I thought maybe a couple of weeks after my kids were diagnosed, that they were up to the letter P and they would do PXE as the next disease. And we know that we're going to have to advocate for that disease, but at the same time we realize we're going to have to do that in a very broad-based way, and not in a way which, for example, would ask Congress to earmark specifically for this disease, but instead find ways that the pathways and mechanisms that we all care about are taken care of. Which brings me to Genetic Alliance. So moving from founding PXE International, I began to work with Genetic Alliance, understanding that we need to work together. And our initial mission in the early years was increasing the capacity of these disease support groups like mine. In the last couple of years, we've undertaken a huge transformation ourselves at Genetic Alliance and understand that really transforming health through genetics is more of our mission. And we have 3 kind of overarching goals in that arena. And the first is creating environments of openness. And that's very broad and very vague in some sense. We're really looking at what is the disruptive technology, disruptive innovation, that openness, and that could be any kind of open source, open aggregations of data like Genbank. We've seen fabulous models here at NCBI, and we're really looking at how do we push those out into other systems and other places so that we're getting this sort of connections that would bring us to the next, where we look at novel partnerships and advocacy. And by that, we no longer mean just advocates like the people who bang on doors for diseases, but instead all of us working together to advocate for open access, for genetic discrimination, for translation, for therapies and therapeutics, et cetera. And we want to revolutionize access to information overall, and we think that, for example, Genbank's a very big example of that, and we're happy to build on the shoulders of such giants. We founded the Genetic Alliance BioBank after having the PXE bank be running about 9 years so that all groups could use the same structure. At that time, my husband and I were getting lots of other patient groups coming to us saying, could you teach us how to do the same aggregation of clinical samples and physical samples? And eventually we said it really makes sense to have an infrastructure that we can all share. And so in 2003, we founded the BioBank with both a number of advocates from disease groups and also advocates from other arenas, including Claire Driscoll from the National Human Genome Research Institute. The BioBank has 7 member groups, and when I say members of the BioBank, it means these disease groups, so things like Angioma Alliance, inflammatory breast cancer, Joubert syndrome, et cetera. So for the most part, they're rare conditions, although we do have even some non-genetic looking conditions like the National Psoriasis Foundation, and that is certainly not a rare condition, so the bank is really able to be quite expansive in how it's functioning. Our vision is really to create a community of trust that worked very well with PXE International. It's more and more the norm for overall research in general, to gather data, to be able to annotate it and manage it in a very professional and controlled kind of way. Open access for that data, incentivizing collaboration and collectives in research, and pushing through to clinical studies and therapeutics and treatments. The problem is that if we want this set of well-annotated samples with longitudinal follow-up and in order to catalyze research, we really need to build a structure, an infrastructure to collect this clinical data and the biological samples and allow for this long-term follow-up, dynamic reconsent, web interfaces, et cetera. And essentially what we were about was breaking down silos as we went. So the BioBank is advocacy-owned, and in this case I mean disease-specific groups-owned, repository for clinical samples and data, and it's centralized. It allows all the groups to collect in one infrastructure. It allows the highest participant protection, so we've always been very careful to follow the highest bars, the highest standards, and the same thing for the repository. It enables ethical recontact, which is often very problematic for scientists and groups that de-identify data, and allows follow-up for genotype-phenotype correlations, natural history, and longitudinal studies, and then open access to the degree determined by the managing organization. So each one of those disease groups that I just showed you, they decide what does the application procedure look like to get access to the data and the samples in the repository. This allows the organizations themselves to be stewards of this entire collection and also to engage in whatever level of benefit sharing the researchers they're working with will allow. The sample, as I said, contains clinical information, DNA, RNA, cell lines, medical records, self-reported data, tissue, organs, whole harvests from autopsies, et cetera. Our first phase, which was quite simple, it was the biological sample repository. Almost anybody who has a freezer on a backup can do that, and I'm being facetious, but it's certainly much, much less complicated than doing the clinical data repository piece, which we launched in 2006. And the third phase was to allow a web interface for donors, for participants in research to be able to put their information into the various questionnaires that we have all developed, and we just launched that this past month. Essentially, it functions like a cooperative. So the BioBank board builds the best possible infrastructure and is continually looking for better ways to do what we do. As the number of members in the BioBank increase, just like any co-op, there's more resources and we can have more options. And then the members themselves bring various skills and resources to a general common structure that feeds directly into Genetic Alliance's many tools, our wikis, our resource repository, our interactive guide for advocates. The system is pretty straightforward. It allows us to be able to put a request in of some kind, and that request could be either for a sample to go out or come in, for information to go out or come in, an online questionnaire for the people whose samples we're collecting, for them and their physicians to answer, or for them to send in medical records and us to enter data. An internal system, we're using a proprietary system called TRIMS. Gene Logic in Gaithersburg built this system. A staging site that allows us each evening to do just general quality control checks of the whole system in a very typical manner, and then for us to manually release or not release certain records, and then all sorts of specialized sort of clinical data entry forms and ways to search the database. Again, the vendor is Gene Logic on this system. Pretty simple and straightforward on one hand, and on the other hand, we looked at a lot of systems and had a very hard time finding something that would be flexible enough for the different diseases that we wanted to put in. Very big step was getting this online questionnaire together, and another, I think, very big step will be when we connect it to the SET structure, which is the Collaboration, Education, and Test Translation program that NHGRI and the Office of Rare Diseases runs in collaboration with NCBI, and that our data is actually captured and transported between the two systems easily. We have an administrative portal where we can manage all of the donors, patients that come in and need to answer these questionnaires. The patients themselves can answer the questionnaire all in one sitting, or they can take several fits and starts to do it. It saves like any other system. It's customizable, so the advocacy group, psoriasis, for example, is doing a study with 2,000 individuals right now, and they've customized the questionnaire to contain questions that are very important for the study that they're looking at right now. And then we look at everything before it gets uploaded to the main database. We're able to collect clinical data on medical history, signs and symptoms, surgeries, et cetera. We also can manage all the physical samples. We have the genotype information in there as well. And then we're able to have a number of ways to release the data. This allows us also to have a workflow that's audited, and we're ready if the day ever comes when we have to go to GMP or to other ways of bringing a drug to market. It's a very controlled system. It uses SNOMED as its key vocabulary. We're looking at upgrades to that as well, since that may not be the best vocabulary for us to use. They've developed other vocabularies that seem to have good interoperability between them and other systems. And then we can get reports out of all sorts that we might need over the year, over the week, or over the month for whatever sort of project that we're working on. It has all the usual security and authentication sorts of things to make sure that we're compliant with the various things that we need to be. One question that's very important to the advocacy groups, and the reason why they haven't just decided either to bank with a bigger system or to leave themselves in some researcher's lab, is how much does this cost? Because while many of the, especially genetic diseases that are more rare, more Mendelian in origin, are frustrated by the fact that their samples sit in somebody's lab for a long time and they can't aggregate aggregate enough samples and they can't get to the research. The reason they never leave that sort of situation is because of the cost. And when we look at typical biobank costs, at least the ones that are made public, we look at the autism one that's 400 families for $6 million, or 1,000 motor neuron disease patients for 750, or a 1,000 sample repository for multiple sclerosis that costs $2.5 million. Our biobank costs $20,000 a year. And again, this is because we've decided that we're going to work with one another. We're going to have the opportunity to share our commons, and will allow then individual advocacy organizations to take care of a lot of the stuff they might have to go off and find themselves or pay for themselves to be paid for through the infrastructure that we've built. And so a typical year for a biobank member collecting 1,000 questionnaires, 100 tissue samples, 100 blood samples, extracting the DNA, blah, 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 is about $37,000, which is fairly reasonable. And even some of the smaller groups, certainly not the smallest, but some of the smaller ones find that quite reasonable. It's important to see, is this useful, is it worth it? And certainly for our group, PXE, it has been, and I'm going to walk you just a little bit through some of the things we were able to do because of this bank. In addition, CFC, which is another one of the rare disease organizations, has gone on to do drug development, and so for them it has been very valuable as well. Psoriasis just started its collection, and so we won't see any results probably till later this year. PXE International, as I said before, is a condition that actually causes calcification, mineralization of some elastin in the body. It has a lot of different phenotypic characteristics, and so it's fairly complex, but every one of those seems to also have a corollary. For example, in eye disease, in macular degeneration, or in cardiovascular disease, in the usual cardiovascular disease. We keep hoping that, since wrinkling is a very common attribute of the disease, that Congress, who seem to be wrinkling a lot lately, will become interested in the disease. The gene for PXE is ABCC6, which is in the ABC transporter family. CFTR is also in that family, and we use this as a good example to the other advocacy groups of, you're certainly always very interested in going down your own path, and in our case, everybody was going down the elastin structural genes path. And when the gene was found, in fact, it's in this ABC family. CFTR is a close cousin, totally unexpected, and a whole different ball of wax. It's a liver disease, it turns out. And so we're really happy that we stayed fairly open-minded about this and didn't follow just one path, understanding that science is often really wonderful in its serendipity. It has a lot of homology with the other genes in the ABC family. And there's a lot of human diseases that are in the ABC family. We just came back from a ABC meeting in Innsbruck that happens every other year, and actually we're able to have those basic scientists do a clinical day this year looking at CFTR and ABCC6 to understand how these diseases might actually be related to one another, and whether, if we study the pathway of one, we can understand for another. We've been very involved in aggregating all of the mutations that we can find, and we find many, many mutations, many of which are novel, very much like CF. We find certain hotspots for mutations, like in these Walker motifs, that maybe will be good at some point for therapeutic interventions, et cetera. But right now, we're very far from any therapies. Causes papular lesions or plaques and the various flexor areas of the body, and then eventually this lax and redundant skin in the body. It causes calcification in what's called the Brooks membrane in the back of the eye. That causes breaking, causes these lines in the eyes called angioid streaks, which allow blood vessels to grow through them, and then people wind up with these very large areas of disciform scarring, very similar to macular degeneration. The good news is recent drugs like Avastin and Lucentis, which are being used for macular degeneration, are also being used in PXE, and they're quite successful, which is very heartening for us. Our kids were diagnosed at around ages 8 and 6, and they're now 18 and 20, and people generally lose their vision around age 30 or 40, so we feel like we have only another decade or two to race against the clock here. We were really interested in understanding, how does this disease progress and what's the phenotype look like? So we put together this tool called the PhenoDex. We took the various attributes of the basic organ systems that were affected, and we decided to quantify them. As you see here, very simple, very simplistic, but some way to actually decide what does this phenotype look like, because it's fairly systemic. We took all of that data, and with the help of some epidemiologists from CDC, did a genotype-phenotype correlation and came up with nothing. And so we instead turned it into a binary phenodex. Essentially, is there any eye sign or not? Is there any skin sign or not? And in this binary system, we were able then to look at that against the genotypes that we aggregated, and we found that in fact there was no statistically reliable correlation and that all p-values were well above 0.05. And so at this point, we have no clinical phenotype. We believe that perhaps there'll be a subclinical phenotype, or that if we look at more, our power here is low. We only had about 250 genotypes up against our phenotypes, and so we're hoping as we aggregate more, we may be able to find some correlation. All of this led us to believe that we really need a systemic approach or a systems approach to disease research, and this is a fairly complicated diagram. But on the left, we're looking at the social network. So in our case, the community of trust that we built. And on the right side, we're looking at a small world network, which is the network of researchers and the network that Genbank and other NCBI tools are really very much engaged with, and in fact are sometimes the glue or the conduits through which these researchers connect themselves. And then using those two networks, we allow the bottom half of this screen to happen, which is the whole research enterprise and various pieces of it that I just talked about, to hope that someday when we get these consortium together and they get working with each other, we will be able to find the therapies and treatments that we all long for. And I think we're coming to a really crux time to be able to do that, as long as we're all willing to kind of push the envelopes that we work with. So one of the envelopes that I'm really concerned about is ownership, and I flippantly here say, get over it. I think we really have to look at consent issues and essentially put them to bed. Figure out, how do we consent people in this day of Facebook and MySpace and wikis, et cetera. We can talk to each other very quickly and constantly, and we simply need to figure out how can we do dynamic consent, how can we make sure that people's privacy is protected as much as is allowed, because I don't truly believe we can protect it completely, and that in fact people are just feeling comfortable because they are in communities of trust when they move into research. I think we need immediate public access. I'm very happy yesterday when it was the first day of the mandated 12-month embargo deposit of information, but I think we need to go to 0, and I'll continue to work on that. I put that in my thank you letters to all the senators and Dr. Zuhuni, and I realistically know we may get to 6 or 3 months, but I think we really need to move quickly in that direction, because the more people can play with this data and look at the information, the more we'll see that we can make strides. Again, I'm a mom with a master's in religious studies. My husband never went to college, and we've been able to do all this because we had access to the data one way or another. Data sharing is going to be critical, and looking at how do we create scientific and community commons that are really democratizing science and allowing all of us to work together with one another. All of this is going to lead a great deal to a lot of policy issues. Public access certainly being a huge one and a big one. Intellectual property issues around how do we safeguard enough intellectual property to incentivize science, but not so much that we're actually creating barriers. And certainly discrimination in insurance and employment is important, and something that I've been working a great deal on, and Francis touched on in his talk. So at the end of the day, I think what matters is our health, their health, and global health. And I really want to celebrate with all of you the disruptive innovation of Genbank. Thank you. I think you're back on time.
Do we have any questions for Sharon?
Yeah, I have a couple of questions. As someone who used to work in the pharmaceutical industry, the patents on genes, that's only the first step in a very, very long and expensive pipeline. And as long as one has freedom to operate through licensing the IP from you, that's perfectly fine with us. My second question is in Tom Ferguson's white paper on e-patients. He talks a lot about the power of social networking. And I've noticed that over the last years, there have been a couple of commercial social networking sites launched, Talmed, Trusera. What role, if any, do they play in all of this? What do you think about that development?
So, great questions. For the first one, I think it's pretty simple. I agree with you, and in fact, holding the patent on a gene, myself, I certainly understand the issues, and I do think it's about licensing, it's about how to be creative about sharing intellectual property, and not about the patent so much. So I agree with that. The e-patient thing, and God bless Tom, his contribution to understanding that field, I think, was way before this, what I think is a tsunami coming at us. And I've been actually talking to the other advocacy groups, and part of why Genetic Alliance is looking at how do we really become relevant and important for moving systems in the new age, is because these groups are actually going to become very, very popular. They're the new tsunami for us. They're going to wash us over, us old brick and mortar advocacy organizations, and we're not quite brick and mortar either. We're internet connected. But you see the kind of popularity of Facebook. I'm on the Google Health Advisory Board, Google Health, Microsoft Health Vault. Those things are going to start to let people aggregate themselves the way Angie's List and Craig's List and all these other things have done for other industries. They are going to start to be the way people find themselves. And I think what's going to happen is either advocacy organizations, the traditional ones, are going to have to morph into groups that are able to take advantage of sort of the downstream of these groups, of these people aggregating themselves, or they'll go out of business, and you know, that's certainly probably fine. My husband and I keep saying our job is to shut the lights off and go home. So if somebody else could figure out how to aggregate people better than we have, great. And if we then, I think the big push should be, so what does, for example, the databases at NIH do with the data that's going to be coming out of 23andMe and Navigenics? And you know, we can like them or not like them, they're going to be there. And if people have their sequence in their hand and want to share it and are willing to enroll in large cohort studies and other things, how are we going to manage that as a nation, as a society? And then looking at that globally, what does it mean for knocking down the walls and silos between countries that have been disruptive in a bad way for us?
The priorities for the competing member organizations, you are an alliance, but I'm curious as how the Genetic Alliance sets its strategic priorities accordingly on the Hill.
Good question. So we are not a typical membership organization. We're not also a 501c6 or whatever it is. We're a 501c3. So we set it through community participatory engagement. So instead of, for example, the groups in our organization voting, it really is, and in fact, part of also why we're not focusing on advocacy groups anymore is our network is enormous. We have 10,000 organizations registered with us who are from industry, government, social science institutes, et cetera, and they're all interested in a lot of different things. And so basically what we do is look at all that information, the board of directors, and then lots of think tanks around us work together to decide what rises to the level of our being able to be effective. So a good example is stem cell research. We've not taken an enormous step forward in that because there are good stem cell research coalitions that are doing a great job, and so we just joined them. Open access, we took a very big front seat because we represent so many people. You know, small land-grant colleges come to us, nurses come to us, occupational therapists, in addition to all the patients. That rose really, really high. So we really differentiate by where can we make a very big impact for the people that are in our broad community.
Thank you. Just one follow-up question. Your budget is, this is a public-private partnership, so where is, if I may borrow a term, corporate America on your work?
Yes, so 1% of our funding comes from corporations. I think that's appalling, and I should bring that to about 33% is my goal. About another 4 or 5% come from foundations. That's appalling. That should be 33%. And most of our money comes from federal grants, mostly from HRSA and CDC. We don't receive any NIH funding.
Okay, well, thank you very much.
Thank
It was
you.
terrific.